So let me thank the organizers for giving me this opportunity. And I've also taken the liberty of altering the title a little bit, and I hope you won't mind. This is the roadmap. I'm going to talk to you about the genographic project which we were involved in. I'll talk to you about Watson for Genomics, and I will conclude with some other different efforts that we are involved in. So most people don't realize that we in IBM Research were amongst the first to dabble in direct-to-consumer genetics. What it means is that you mail in your DNA and you get some interpretation, some information from it. So of course so this is my genealogy that I'm very proud of, and here are some of my ancestors. Okay. So I get a kick out of this because I'm ordinary, but my ancestors are extraordinary. But of course I didn't mean this genealogy. This comes from the American Mathematical Society. And I didn't have to mail in my DNA, just my ID, and I got it. And I'm sure you probably have in your own areas your own genealogies. But this is the DNA, genealogy based on DNA is what I meant. And I actually am fascinated by the fact that my DNA just not determines the color of my hair, skin, height, state of health, disease, and so on, but is also a history book. It's a palimpsest of the story of all my ancestors, going all the way back in time when we as Homo sapiens were just hanging out in the savannas of Africa. But of course, if you look at my DNA alone, you cannot get this whole history. We have to look at the DNA of multiple individuals and reconstruct this common history. Not ancient DNA, but individuals of today. So this was the inspiration behind the Genographic project, and it was a collaborative effort between National Geographic, IBM Research, and a whole lot of other PIs from all over the world. And in this project, we managed to gather more than 800,000 samples, and this community, more than anyone else, realizes the significance of data, nothing to sneeze at. But today I want to talk to you about some of the science results we got from this project. And when I talk about this, people often also ask me how we got what results we got. So I'll spend a little bit of time saying how we got what we got. And of course, all of us know that the Y chromosome tells the story of a man, his father, his grandfather, great-grandfather, and so on, which is the purely paternal lineage. And mitochondrial DNA tells the story of a woman, her mother, her grandmother, great-grandmother, and so on, and which is the purely maternal lineage. And the common history, turns out, is a clean, wholesome-looking tree, as you see here. But this has ignored my paternal grandmother, my maternal grandfather, and a whole bunch of other ancestors that I have. So the question that we asked is, if we look at all the lineages, can we not look at all the lineages? But it turns out that the common history is much larger and much more complex. And this is called the ARG, or the Ancestral Recombinations Graph in the population genomics community. And we thought we should try to reconstruct this. But of course, the first question is, this is such a complicated structure, it's so large, can it even be reconstructed? Or what percentage of the ARG can we reconstruct? And we tried to set this up in a way that is answerable. So assuming a baseline that you can reconstruct 100% of the tree, what percentage can you reconstruct? But of course, we can take a community effort, a RECOMB-like community effort, have one team reconstruct one ARG, another team reconstruct another ARG, and ask the question, whose is bigger? And that is a totally valid way of doing it. But one of the ways we decided to do is we tried to answer, because this is so clean mathematically, at least you see the structures mathematically, we decided to answer this question theoretically if possible. By the way, what you see on the left, the clean tree results from real data. It's based on mitochondrial DNA from about 500 samples. What you see on the right is a little toy example of less than 50 individuals of a very tiny segment, just to contrast the complexity. So let's go to the origin of the subject. I will not take very long and go through the entire history. And I'm showing you Darwin on the left and Gregor Mendel on the right. And there is a book called Brilliant Blunders by Mario Livio. And if you have not already seen it, it makes a good read. It talks about mistakes made by brilliant scientists. And Darwin undoubtedly was the most brilliant minds, but he made a small mistake, and this was regarding inheritance. So roughly speaking, what it means is that if you have a blue homozygous father and a red homozygous mother, in F2 generation, the second generation, all the children will be purple. This was Darwin's blending rule. Whereas Mendel, he had taken a peek at the peas and so on. So he said in the F2 generation, you'll have some blue progenies, some red progenies, and some purple progenies. So hold on to this thought, because we will come back to this later. So here is a picture, a meme. Just try to remember this. And this is from textbook 101 in population genetics. So the matrix that you see, each circle is an individual. The bottom row is present. The top row represents the past. And the edge between the two circles denotes the relationship between a parent and child, or the transmission of genetic material. And now if I take those magenta units from the bottom, that is 4 extent units, and try to trace their pedigree, trace their ancestral material, and shake it out, you get a tree that you can see there. And this has a property that you can find one node that is called the least common ancestor in computer science, or most recent common ancestor in population genomics, which is ancestral to all the 4 leaf nodes and is the least one. That is, it is not ancestral to any other common ancestor. You may have noticed that this is a simple example where we have taken only one parent for child, but of course you can take multiple parents and you will get multiple LCS. So these structures are called DAGs, directed acyclic graphs. Although I did not show you a direction here, it doesn't matter. You can either take, so I'm following the convention of downward direction. You can take the other direction. As long as you're consistent, everything will be fine. Population genetics traditionally was born in the departments of statistics and is continued to be studied in the Department of Statistics mainly. We are taking a random graphs or random DAG viewpoint to this problem. So now I realize that we are close to lunch, but let's see how many of you are actually awake. So I'll give you a little puzzle here, where you remember the meme that I showed you. So instead of the 4 magenta units, now I have these 4 hollow circles. The blue solid ones represent males, the red hollow circles represent females, and you can see that the pattern between generation 2 and 3 exactly the same as between 3 and 4, exactly the same as 4 and 5, and it goes on ad infinitum. What I'm showing you is an unbounded arc. And my question to you is, where is the LCA? The least common ancestor or the MRCA? Where is the LCA? I guess you're more familiar with the computer science terms. I do not have too much time, so I will give you the answer. But I'm sure given time you will know what it is. So these are the ones with these double circles. Okay? This is an LCA because it's ancestral to all of them and no other common ancestor in its path. Similarly this, similarly this, similarly this, and ad infinitum. And this leads to a paradox. Why? Because these 4 units have a finite genome but have infinite number of LCAs. So there is a problem. And I call this the ancestor without ancestry paradox. And it comes from the fact that if we try to track the genetic elements in, let's take these 2 individuals, this one is actually, by looking at the DAG, it is an LCA. But if you look into them and what is the genetic element they're carrying, they're ancestors, but they have no ancestry to the genetic material in these. And now I connect back to Charles Darwin and Mendel. If Darwin were right, this wouldn't have been a paradox. This would have been fine. It's because Darwin was, it's not because Darwin was right. So we can make some simple assumptions and can come up with paradoxes. And because Charles Darwin was not correct, it was not the blending theory, but the mixing theory, this turns out to be a paradox. So the point is that what looks like an arc and what talks like an arc is not necessarily an arc. So we sort of built what an arc definition should be, just enough to show that this example that I'm showing you is an arc because you can actually have internal elements in it and track them The next thing that we did, because we do want to answer the question that I had posed, okay, so I'm just setting the framework for that. What we did next is we tried to mathematically identify the core, a minimal descriptor, a minimum core that contains all the information about the arc. In this toy example, perhaps it's not clear, but the second picture there is a little smaller than the large picture. This is a toy example, so the reduction is smaller. And after a couple of theorems like, even bounded arcs have, unbounded arcs have bounded minimal descriptors, and the minimal descriptor is unique, we have actually answered the question that we set out to. That this gives a theoretical upper bound on the extent of reconstruction. That is, no matter, this is assuming no false positives and other things, no matter what algorithm you have, the best algorithm cannot reconstruct an arc which is bigger than the minimal descriptor. And in practice, you are always reconstructing smaller arcs than the minimal arc. And there were other consequences, but I'll skip that. And so this is the question we answered, is that this is through, this is algorithm independent. Of course, you want to make it algorithm independent, then you have actually answered the question. And with simulations where we use simulated data, a third-party simulator, we didn't use our own simulation, third-party simulator where we took populations that reflect the molecular profiles of current populations at a broader scale, like African populations, Asian, and so on. And we concluded that 35% of this arc is permanently lost to any recovery through only DNA. This is the common history, the arc history. So I see Manuel is frowning, and I think there'll be a question at the end, but and of course, the 65% is kind of large enough, and so we designed some algorithm to reconstruct it, mainly using graph theory and so on. So what's on the right is the theory, and I'm saying on the left, what I'm showing on the left is practice. What it means is that you have to account for uncertainties and so on, so calibrations, different parameters, and so on. And when this at the time this algorithm was designed, when we published it, it was the first algorithm to reconstruct the arc at a genomic scale. And how we did that was actually using pattern discovery and other ideas from strings, brought them into a graph theory to actually reconstruct this arc. So we take the genographic data that we have, and then we construct the arc. This is a transient arc. What I mean by that is that due to uncertainties and so on, you actually build multiple arcs. And of course, it's very complex, and you don't know what it means, but you can actually check different hypotheses against this structure. And one of the striking results from this was that based on this data, based on the analysis, the result is that all of us, there is consensus in the community that we as humans originated in Africa. And the question is, which is the route that we took when we left Africa? And our analysis actually favors the southern migration through Bab al-Mandeb over the northern migration through Sinai. In fact, we sort of redrew this out of Africa map along with some deep incubation point in South Asia. And since then, there have been other papers which have corroborated these results. I kind of like to put this up. IBM had a 100-year celebration, centennial celebration a few years ago, 5 or 6 years ago. And they have a time capsule now in the lab, and there is a genographic project kit in that lab. So I really don't know whether mankind will lose all recorded history and will not know anything, and they find this capsule and then they'll see these kits and other things. I don't know. I just find that amusing because I think recorded history will probably keep a lot of them alive. So moving on to the second topic, and now I want to talk about Watson for genomics. This is about cancer, and cancer is a disease of the genome. I don't need to tell this crowd, but this is a new theory. What we have learned about this disease in the last 50 years is a lot more than what we knew about the disease in the last 5,000 years. And this is a question I ask. Did the disease exist 5,000 years ago? Yes. It actually did, and we know that because we see that in the mummies from the pyramids in Egypt. So the disease did exist, but it's only in the last 50 years that it got established as a genomic disease. And also, like Dana was mentioning and everybody also knows, is that no two individuals' cancers are identical. So this makes it a nice candidate for precision oncology or precision medicine, because it's the same disease, but it differs from person to person, and you can actually target individuals. And again, you probably know better than I do that in literature we are seeing evidence that when you have molecularly targeted therapies, it improves the lifespan of some, if not all, patients. And the question we would like to ask is, what will it take to give every patient the opportunity for an exceptional response? Not to the patients who have access to, say, like top cancer centers like MSK or in NYC or Boston or LA, but in every corner of the country, possibly every corner of the world. And so we built what is called Watson for Genomics. We call it Watson for Genomics, but its focus currently is on cancer, because cancer is a disease of the genome. And you may have seen its coverage in 60 Minutes by Charlie Rose, when he was actually did a piece on impact of AI on society and community. So Watson for Genomics was covered there. Interestingly, Watson for Genomics started as a very simple exercise in our lab, in the research lab. And then it grew, and now it has moved to our business unit, and now it is a commercial offering. So I'm afraid I'm not at liberty to talk about the details of how it works, but I will share the challenges. I'll share with you the challenges that we faced, and I hope you'll find that interesting. So just to recall here, when you take a patient data, it is the patient's molecular profile, it's DNA-seq, epigenomics. For the moment, we are taking only DNA-seq and RNA-seq data wherever they're available, because this is a practical situation that we're dealing with. Not all centers and not all hospitals will give you RNA data, so it might be sometimes just with DNA data. And the fact that the size of data per patient now is pretty large. So the first challenge you face is the existing knowledge in unstructured format. Unstructured format meaning in terms in literature, in papers, and so on. And for this, we use the engine of Watson that you probably already know about, which played Jeopardy on national television a few years ago. And this handles the explosion of knowledge that we have both in the structured space and some in the unstructured space as well. And this actually gives is a good fit to this data's velocity, volume, variety, that the 3 V's that people talk about, big data, and cognitive, meaning AI computing here. So of course, this is not the same as playing Jeopardy. So we have to train the system to understand literature in this context. So you plan, you build text extraction, you build the medical logic, and you iterate through and improve the system. The second challenge is, of course, the genomic connection. So the question is, we are talking about the system today, here and now. Of course, tomorrow we will have a better understanding of the disease and better ways to fight the disease, right? And so the question now is, what must a system, not just Watson for Genomics, any system, how much of cancer biology must it understand to be effective to society? And we know that as cells divide in our body, mutations are accumulating. Most of these mutations are harmless, but sometimes they're not. And the state of the cell goes from precursor to benign to malignant to metastatic. We don't understand why or when this happens, but it does leave a footprint behind, and a system can actually utilize that footprint. And it needs to know the difference between the current theory is there is a driver gene or there are driver mutations, so it just needs to know the difference between drivers and passengers, needs to know why. because the current therapies are mainly targeted towards the driver genes and driver mutations. And also the difference between oncogenes and tumor suppressor genes. Again, why? Because these leave a different kind of footprints. And the summary of this thing is not to tell you what is the driver gene and what is the tumor suppressor gene. It is to tell you that a system like this can take some simple definitions or make some simple knowledge assumption and can be somewhat effective. And of course, the challenges here is that disease is very heterogeneous. It differs from person to person. Within the same person, it differs from tissue to tissue, organ to organ, and within the same tissue or the same organ, it differs from location to location. And some patients have a small set of strong drivers, and some patients have many weak drivers. And there are some cancers driven purely by mutations, mainly by mutations, such as kidney cancer. And there are some cancers like, say, ovarian cancer, which is driven primarily by copy number variations and so on. So these are the kind of heterogeneity that it needs to deal with. And the fact that it is not just the driver genes, it also tries to reconstruct the pathway that it believes, the disease pathway that it believes that underlies that patient, because therapies can be targeted towards downstream genes as well. And of course, it needs to know what are the drugs there, because it's, it is actually something that we want it to be useful directly at the other end of being useful. So we want to know the cancer trials. It needs to know about resistant and sensitive markers of the patients and so on. So to summarize, Watson Genomics will take the patient omic data. It has ingested the unstructured literature and some structured data as well, like COSMIC, DB-SNPs, Ensemble, and such. And it outputs two important lists. One is an ordered list of driver genes, what it thinks are at play for that patient, and the other is a list of drugs, again, which it thinks will be effective for the patient. And also it gives circus plots, it surfaces what it thinks are the important mutations for the user to take a look. And the pathways, again, this is a simplified disease pathway. It is a hypothesis of the system, what it believes is at play for that patient. There are many ways to measure accuracies, but of course, the top of our list was the accuracy of the precision and recall of the driver genes and the drugs. But how does one do it? Because, as you know, there is no benchmark data in this context. So we worked with early adopters and partners, and thanks to their patience and cooperation of working with us, and we discussed every case by case, we discussed this with them, and we went from Watson for Genomics giving nearly laughable results to, shall I say, not laughable results. And that's when rest of the organization begins to take you a little seriously, and that's how it moved out of our hands into a business unit there. And here is an example let me give you. So this is with BCCA, British Columbia Cancer Agency. This is one particular patient for lung cancer. We both agree on the molecular signatures for that patient. And here are the drugs that we both agreed on, but these are some of the drugs produced only by BCCA and some drugs produced only by Watson for Genomics. In the two drugs, in this particular case, this is one specific case that we pulled out at random that I'm sharing with you. The top drug that Watson for Genomics did pull out was a drug associated with chemotherapy, but that's outside the scope of Watson for Genomics. And the second drug that Watson for Genomics did pull out happens to be, it turned out that there was a resistant marker in the patient which the system had seen. So in some sense, it not pulling out both those two drugs were justified. And the drugs that Watson for Genomics pulled out, these are retrospective patients, so the drugs that Watson for Genomics pulled out was considered, the BCCA counterparts acknowledged them as valid drugs for that patient. So to summarize, of course, it's comprehensive, transparent, and navigable. What I mean by transparent and navigable, I probably didn't mention, is that the user can actually click on various things, sort of ask the questions as to why does it think that these are, why does it give the answers it gives, and it is totally transparent, and it will tell you the literature or what is the reason why it has given these answers. Of course, it's a system, so it is scalable. You can go from tens of patients to thousands or millions of patients, and the turnaround time that we compute and we write in the papers is 6 minutes, 6 to 10 minutes for the system, whereas for the humans, based on different cancer centers, it goes from 6 hours to a couple of weeks to even months. So the turnaround time is really quick for a system such as this. And the other thing which is extremely interesting is that it is updatable. It can be made state-of-the-art current by simply pushing a button. That is based on the literature and other things. It can, with the push of a button, it may not take an instant, it will not update in an instant, but all you need to do is push a button and it is updated. And the fourth one, which is again very interesting, is that it has the potential of giving you something new. That means it can potentially augment a clinician's list of possibilities. But again, it is transparent and navigable, so the clinician, it just assists the clinician. It does not replace the clinician, but the clinician can actually go back, query, and see whether these things make sense or not. So now, I want to share with you something that we got from a third party. What I mean is that this is kind of a user that we're using a research version, and this is something that appeared in the media totally unbeknownst to us. And let's watch. This is a good piece of video, so I'm sharing it with you.
Artificial intelligence is rapidly bringing big changes to life as we know it. It's been used in self-driving vehicles, beaten chess champions, and is now helping with medical diagnosis. Doctors in Japan are touting a breakthrough, saying AI has saved a patient's life. NHK World's Tomoya Oyabu has the details.
Last year, Ayako Yamashita was diagnosed with acute myeloid leukemia, a type of blood cancer. For several months, she went through treatment using anti-cancer drugs, but her condition worsened, and doctors couldn't figure out why.
I wasn't sure whether I could live until the next day or the day after that. I didn't expect to live to see this day.
It was Watson, a computer system developed by US IT giant IBM, that saved her life. Researchers at Tokyo University's Medical Science Institute are conducting clinical studies with IBM. They want to see how Watson can learn about cancer and diagnose the disease.
It stores data on over 25 million medical papers, as well as over 15 million drug patents. It has information on the mechanisms of life.
There are different types of leukemia, each requiring different treatment. Doctors at the university used a supercomputer to analyze alteration of Yamashita's genes. In just 10 minutes, Watson concluded that she had secondary leukemia, and it proposed that doctors use a different anti-cancer drug.
I believe only artificial intelligence can present related data and reasons behind a diagnosis with that speed.
Yamashita responded well to the new treatment and was discharged from hospital.
The help of artificial intelligence, in addition to the doctor's treatments, doubles my sense of assurance.
Doctors say Watson provided helpful information to treat a total of 41 patients, including accurate diagnosis for 2 cancer patients. They say AI technology is opening a new horizon in the field of medicine, beyond human intellect. Tomoya Oyabu, NHK World.
So we are happy for this patient, and we hope that we will have many more such cases. And now Illumina also has added IBM Watson report to its 170 panel genes. And of course, until now, what we are doing, if I may call it, it was the imitation game, in the sense that we were trying to imitate what is already known and what is happening, we were trying to put it into a system. So we were playing the imitation game, so we were imitating the molecular pathologist and the clinical oncologist and so on. But of course, we also want to understand the disease. We also want to understand cancer. And so there's an effort with Broad. Now, I know many from Broad are here. This is with Gadi Gatz as a PI from there. And we are trying to understand drug resistance, as to why do certain patients become resistant while other patients don't. And hopefully, we hope that in the next few years, we will have some nuggets of wisdom there which we can feed back to Watson, so that it can help, it can assist the clinician with what drugs the patient may become resistant to. But this is something ongoing, and hopefully we will get there someday. So a few weeks ago, actually, I was having a chat with a long-standing colleague, and he lamented to me that there is a loss of diversity in the class of tools that we are applying in the world of big data. Of course, statistical learning, machine learning, I mean, who can resist their charm? Of course, all of us want to use it. There's evidence in the morning session itself. Almost every talk had some machine learning thing, and it should be used. So the next part of the talk is sort of inspired by this conversation, his worries or his concerns about that, to say that there are other kinds of tools as well. And so in that context, so that is kind of dedicated to my friend Gadi, who is not here. This is not Gadi Gatz, this is a different Gadi. Because I kind of wanted to answer his question and say that there are other things also, some old tools, new tools that one also looks at. And in this context, I want to talk about three streams. I will go very quickly over this, not to worry that you will miss your lunch or something like that. And I'll also talk about what are the tools that we are trying to use here. So the first one is the health conceptual shift. So there is a DNA that we are born with, and now it is becoming we think that the second genome, that is the DNA of all the microbes around us, the microbiome in our guts, on our skin, in every pore of our body, they probably play an important role in our disease and health state than we had earlier anticipated. And this is being called the second genome. And the number of the microbes is almost the same as the number of our cells, which is approximately 40 trillion cells. Our first genome has around 20,000 genes, and this microbes, they have around 1 million genes. And that's the reason why people think that it probably plays a more important role. And the gut microbiome is being called the linchpin. It's being called the neurological control. It's a second liver, it's a second endocrine, it's an immune trainer, and so on and so forth. So the question we are asking is this gradual conceptual shift as a human, what am I? Am I a closed system or an open system like the Amazon forest and the corals and so on? And the human medicine, also there is this shift from this aggressive war metaphor of the last century, like annihilate, destroy, and so on, to this gentle gardener metaphor of nurturing and tending our microbiomes and so on. So this influence of microbiome in the state of disease, health, and aging, is it taking it by storm, or is it a storm in a teacup? We don't know, but time will tell. But this is something that more and more people are paying attention to. There are clinical trials which are being designed around microbiomes, and it is gradually playing an important role there. And what are we doing here? So this is metagenomics, and I specifically mentioned this here because in metagenomics, for people who are not familiar with metagenomics, what it simply means is that if I take the DNA of everybody in this room, put them together and feed it to a sequencer, out will come the reads, and now the algorithm has the responsibility of separating the reads and mapping it to the different individuals. And this is the problem that we are in metagenomics. That's the problem that we face. Of course, here we mean the DNA of the microbes. And the microbes are not like us. They beg, borrow, steal genes from each other, and they have this horizontal gene transfer leading to confusion and the kind of databases, and that's where the complexity of the problem lies. So here one actually resorts to basically string algorithms. You may have heard K-mers and so on. So there are string algorithms that are in use in this domain. We are also part of the Sequencing the Food Supply Chain Consortium, and this is to do with microbes or microbiome, not in your gut, but in the food supply chain. If you had the time this morning also there was a recall of a corn product due to listeria, and then you might have heard of ice cream recalls due to listeria, or peanut butter recall due to salmonella, and so on. So it comes at a cost to health, as well as it also has an economic cost associated with the recalls. And the idea here is to monitor the microbiome in the food supply, understand the profile, and predict it even before it happens. So this is not to do with the gut microbiome, this is to do with the microbiome that exists, say, in the food supply chain. And this is something that we are working on as well. And again, this is string algorithms, phenotypes, machine learning, and so on, so a whole host of algorithms that are utilized there. You probably never thought that you will see these three words, logic, topology, and genomics, in the same sentence. So anyway, let me try to say what I mean. So at the risk of being a reductionist, I'm going to give you a 2-minute description of TDA, or topological data analysis, to people who are not familiar with it. So this mode of analysis actually has a refreshingly different way of looking at data. It doesn't care about distances, and to a certain extent even scale. So it looks at the topology in the data. So the second thing is the simplices and Betty numbers. What are they? But we want to represent them in a computer, we want to digitize them, we want to manipulate them. So we use simplices and Betty numbers by which these things can be represented in a computer, and you can use linear algebra to manipulate them. And finally, it comes in various flavors. Simplicial complexes come in various flavors like Chinook, alpha, Vietoris, RIPS, and you can map your problem, your data cloud, into one of these complexes based on what your application is and based on what is the question that you want to answer. So the left-hand side is the framework, and in the right-hand side is the actual application of this. So filtration actually means removing the signal from noise. Okay? And persistent homology is something that will go and detect non-obvious features from the signal in your data. So this is my summary of TDA in 2 minutes. But of course, there are many challenges. I have been looking at this problem for a while now. I mean, this area for a while now, and trying to apply it in different areas. And I will probably show you one of the examples. It is challenging to map your problem into this domain in such a way that when you have answers from this domain, it actually is interpretable, and you can go back to your original problem that you asked. Then the second part, let me give you a dirt simple example about what I meant by logic. So these are Boolean expressions. So imagine these are countries with land area greater than 3 million square miles, tourist destination in the Americas, permanent members in the UN Security Council, and those with defense budget greater than 30 billion. If some of these sentences actually some of these are not in agreement, it's simply because I'm using an old slide which I didn't change, because people have told me that budgets of other countries also have grown. The defense budget of other countries have really grown. So you can see that this slide is really dated. It's kind of pretty old. But the point here is, now if I want to define Russia and China, it actually has two descriptions. It is blue circle except the yellow circle, or it is green circle except the red circle, which are called re-descriptions. And how are they related? They could be patients in the data, and we could be looking at different pathways. And it is possible, so in complex diseases, they have complex etiologies, and it is possible that you see a pathway that is associated with type 2 diabetes, and some pathway associated with cardiac disease, and the patients are actually showing both the pathways active in them. So it is a way to understand complex diseases. and to connect it to TDA, we can take these redescriptions and associate them to simplicial complexes and run persistent homology on them to extract characteristics. So this is an example of applying TDA. This is in microbiome data. So this is the data from about 100 individuals. It is a gut microbiome, and red, so there is a prediction of the phenotype here. Phenotype is the state of health of the host, which is red means disease, green means healthy, and there are some other shades in between where it doesn't necessarily have to be binary, so it is somewhere in between. So the card has been flashed at me, so I'm going to now rush a little more. So to kind of summarize this part, so our idea is to use logic, intuitionistic logic to be precise, along with topology to understand complex diseases. And there's an engagement with NIH on this Alzheimer's data, and we hope that we can learn something from the methods on the left that we can apply on the right. Now moving to the last part of this thing, which is using cryptography and genomics. And so the community here, I'm sure, knows about the Hela cells, and the history or story behind it is that this was an ovarian cancer, I think, patient in the 1950s. The doctor biopsies the tumor and then immortalized the cells, and most of the cell lines that you get, the Hela cells, actually come from there. But the permission was not taken from her. Such a thing didn't exist in the 1950s. So such a thing will not happen today. But the question is, what are the other Hela-like things happening today that we have not realized that we are invading people's privacy and so on? Another example I'd like to take is this. This is an episode of 48 Hours, which from on CBS. I don't know if you have seen this episode. If you have not, take a look. It's interesting. Let me tell you what happens here. A crime was committed, and the police gathered DNA from the crime scene, which they believed was that of the perpetrator. But it did not match any of the databases the police have. So they went to a public database. I think that was 23andMe, of course, with a subpoena from the judge, and matched it against all the samples that they had. And lo and behold, it matched not the perpetrator, but a relative of the perpetrator, who they believed was the father of the perpetrator. I will not spoil the rest of the thing for you. There's a little bit of a twist and surprise which you might want to go and take a look. But the point is, we as scientists and individuals, want to donate our DNA for science. But am I jeopardizing my whole family? Is there a malicious user or a state user or something else who it was not intended for, and it comes in? So this raises these sort of questions, and people in the cryptography community, and we work with them, and the cryptographic algorithms, we try to kind of define scenarios of, say, private outsourcing or private cooperation, and we try to define scenarios where you can give your DNA and research can be done on that and so on without actually compromising your privacy. And I gave you a broad spectrum of things. It's not possible without the grit and hard work of my team. I thank my team. And most of the work that I presented also has external collaborators, and you'll find the details in our webpage. And thank you for your attention.